Hey y'all, my name is Campbell Eckhardt, and I am a rising senior at the University of Tulsa majoring in biochemistry. This summer, I had the opportunity to work at the University of Florida's College of Pharmacy in the Department of Medicinal Chemistry thanks to Summer Undergraduate Research at Florida, also known as SURF. More specifically, I worked with faculty advisor Dr. Yusong Ding and postdoc advisor Dr. Dipesh Dikal.
Before I get into my project, I must first explain the basic principles that helps guide my research. Through billions of years, organisms have developed specialized secondary metabolites through selective adaptations in environmental pressures. Now, these secondary metabolites, or natural products, have a variety of uses from chemical defense to mediating organismal interactions. Now, you may be wondering, why does this matter to you? And well, the answer is quite simple. Humans have been using natural products for generations to help improve their quality of life. For instance, aspirin, an over-the-counter anti-inflammatory drug, is derived from a natural product called salicin, which is isolated from the bark of the willow tree. Now, due to their wide range of uses, and as you saw in the example, their importance in drug development, natural products have become economically valuable, as well as the target of global research efforts, including my own here in Florida. Now, there is one key detail I left out of the background, and that is methods for isolating and collecting natural products are far from sustainable. That's where my project comes in. Working with Dr. Dipesh, we focus on natural products produced from strands of marine cyanobacteria. Marine cyanobacteria are known to produce over 1,600 different natural products from biosynthetic gene clusters, or BGCs for short. Unfortunately, cultivating marine cyanobacteria in the lab can provide a challenge. In addition, engineering these strands to produce specific projects has been eluding scientists and engineers. Therefore, we worked on producing a new plasmid that contains the cyanobacteria BGC so that we can express the product in heterologous hosts such as Bacillus and E. coli. Furthermore, we wanted to increase the productivity of the BGC so we're able to produce more product. So not only were we able to produce plasmids in other strands, we're able to increase the efficiency that the BGC works. So, how are you able to engineer new plasmids? Well, first, you need to be able to create a suitable vector. To accomplish this, you first amplify a gene of interest, or BGC, with the polymerase chain reaction. Once you have an amplified gene product, you can then insert the gene into a vector through restriction, digest, and ligation. In these steps, a restriction enzyme will cut open a plasmid, and DNA ligase will then covalently bind the BGC into the new plasmid. Once this vector is engineered, you can then induce host cells to uptake the plasmids through transformation. After transformation, you can then test for protein expression to confirm the BGC was correctly incorporated into the cells.
In conclusion, by engineering a new vector to express marine cyanobacteria BGCs, the Ding Laboratory will be able to increase the efficiency of production of both old and novel natural products. I'd like to extend a special thank you to Dr. Ding for hosting me, Dr. Dikal for his mentorship, and all the members of the Ding Laboratory for their help over the summer. I would also like to extend a thank you to the SURF program for funding, housing, and all those awesome excursions over the summer. Thank you.